Ever wondered how you managed to remember, I don't know, that really embarrassing moment from high school?
Hmm.
Or maybe why your reflexes are just so incredibly quick?
Yeah, those things we take for granted.
Exactly. Well, today we're taking a deep dive into the absolute core of what makes you, you. We're talking about the central nervous system.
And those old analogies like simple switchboard or even a cloud of networked computers.
Right. They don't quite cut it.
Not really. I mean, they hint at maybe some of the spinal cord's efficiency, sure, but they just don't capture the fantastic complexity of the human brain.
Mm. Think about this whole evolutionary process called cephalization.
Uh-huh. The development of the head end.
Yeah, the elaboration of the anterior CNS, that massive increase in neurons in the head. Well, it reaches its absolute peak right here
Like,
in
it
us.
really is quite something, an unassuming powerhouse, you could say.
Totally. Just about two good fistfuls of, well, quivering pinkish gray tissue.
Weighing around 3.3 pounds, give or take.
And it gives almost no hint of its incredible abilities.
Which is why we're so excited to get into this deep dive today.
Exactly. So welcome back to the Deep Dive. We're your personal guides, helping you unlock the secrets of Chapter 12 from Human Anatomy and Physiology, 10th edition.
Our mission today, to give you a real shortcut to being well-informed about the central nervous system.
That's your brain and your spinal cord. We'll
What?
cover the intricate structures, the crucial functions.
And some really surprising clinical applications, too.
Yeah, we guarantee you'll leave with plenty of those aha moments about your own inner workings.
So to set our course, we'll start right at the beginning. How does this incredibly complex system even form in the embryo?
Good question.
Then we'll navigate through the major parts of the adult brain, the big cerebral hemispheres,
Vital diencephalon.
the foundational brainstem, and the coordinating cerebellum.
And along the way, we'll definitely explore those fluid-filled ventricles and the amazing protective layers that basically act as armor for this delicate system.
Okay, so you mentioned cephalization, this drive towards complexity in the head. How does something like our brain even start its journey? From just a few embryonic cells.
It starts surprisingly simply, doesn't it? With just an embryonic neural tube.
Right, very early on. By week 4.
But what's truly remarkable is how fast that tube's front end expands.
Mm.
And it starts to fold in on itself.
Forming just 3 primary brain vesicles at first. Prosencephalon, mesencephalon, rhombencephalon. Forebrain, midbrain, hindbrain.
And then those quickly subdivide again by week 5. The real story here isn't just the names, though.
No, it's the speed. The incredible speed and precision of that early growth. It sets the stage for everything else.
Exactly. That rapid differentiation turns those initial forebrain, midbrain, hindbrain areas into really distinct structures.
Yeah, like that forebrain just balloons out to become the 2 massive cerebral hemispheres.
Right, and other parts specialize into vital areas like the thalamus and hypothalamus.
The midbrain stays relatively small, compact.
And the hindbrain.
That refines into the pons, the cerebellum, and the medulla oblongata, all becoming key parts of the brainstem.
Wow. It really is like a master class in biological engineering, fitting all that complexity into such a tiny, developing space.
And it grows so much faster than the skull around it.
Which leads to that folding, right? It has to fold up to fit.
Exactly. Think of trying to stuff a big, expanding blanket into a small box.
That's a good analogy.
The cerebral hemispheres end up taking this kind of horseshoe shape, growing back and sideways.
And eventually they cover up the diencephalon and midbrain.
Right. And by about week 26, that continuous growth creates all those wrinkles and folds on the surface.
The convolutions. Juri are the ridges, sulci are the grooves.
And that folding dramatically increases the surface area, which means more neurons can pack into that limited space inside your skull.
Which is why the adult brain looks so wrinkled. It's all about packing in that neuronal power.
Precisely. So when we look at the adult brain, we generally break it down into 4 main regions. Those expansive cerebral hemispheres we just talked about,
The big ones on top.
the centrally located diencephalon, the foundational brainstem, which includes the midbrain, pons, and medulla oblongata,
And number 4.
and the cerebellum, tucked underneath at the back.
You know, it's interesting how quickly the brain setup gets more complex than the spinal cords. What's the significance of that change, the gray matter and white matter distribution? Especially when that bark appears.
That's a great point. In the spinal cord, it's pretty straightforward.
Right. Central cavity, gray matter inside, white matter outside.
But the brain adds layers of processing. So in the brainstem, you start seeing extra gray matter nuclei scattered within the white matter.
Handling autonomic stuff, cranial nerves.
Exactly, crucial functions. Then in the cerebral hemispheres and the cerebellum, you get that outer layer, that bark of gray matter we call a cortex.
And the cortex is where the higher level stuff happens.
That's right. Thinking, planning, interpreting senses. It needs a huge surface area for all those neuron cell bodies.
So even though it's packed with neurons, the brain isn't solid all the way through. It has these chambers inside, filled with fluid.
Yes, the ventricles, like little internal bellies, as the name suggests, lined by specialized ependymal cells.
And they're not just empty space, are they?
Absolutely not. They're critical. They house cerebrospinal fluid, CSF, and its pathway is quite intricate.
Okay, walk us through it.
You've got a pair of big C-shaped lateral ventricles, one deep inside each cerebral hemisphere.
Separated by the septum pellucidum.
Right. These connect to the narrow third ventricle, which sits in the diencephalon.
Via what?
A channel called the interventricular foramen.
Okay.
From the third ventricle, the fluid flows through the cerebral aqueduct like a canal running through the midbrain to reach the fourth ventricle.
And that's in the hindbrain, dorsal to the pons and medulla.
Correct. And crucially, the fourth ventricle has 3 openings, apertures.
Letting the fluid out.
Exactly. 2 lateral, 1 median. They connect the ventricles to the subarachnoid space.
Which is the fluid-filled area around the brain.
Yes. It's this whole internal plumbing system, really, for protection and nourishment.
All right, let's focus on the command center itself. The cerebral hemispheres.
The big players.
Yeah, the superior, most obvious part of the brain. They make up the whole top part you picture.
About 83% of your total brain mass, actually.
Wow. And they almost completely cover the encephalon and the top of the brainstem.
And their surface, that landscape, it's marked by those elevated ridges, the gyri.
Separated by shallow grooves, the sulci.
And then the deeper grooves, the fissures, which separate larger regions.
Like the big one down the middle.
The median longitudinal fissure separates the 2 hemispheres, and the transverse cerebral fissure separates the cerebrum from the cerebellum below.
And they aren't just wrinkles, they're functional, packing in more processing power.
Absolutely.
Now, each hemisphere gets divided into 5 lobes, right? By those sulci.
That's right. Frontal, parietal, temporal, and occipital lobes, mostly named for the skull bones covering them.
And the 5th one, the insula.
Yeah, that one's tucked away deep inside the lateral sulcus, almost hidden.
Key landmarks are the central sulcus.
Separating frontal and parietal lobes.
And the lateral sulcus outlining the temporal lobe.
And fundamentally, if you zoom out, each hemisphere has 3 basic regions.
Okay, what are they?
First, that superficial cerebral cortex, the gray matter bark we talked about.
Second,
internal white matter.
And third.
Deep within that white matter, you find islands of gray matter called the basal nuclei, each with a distinct role.
So we've mapped the physical terrain, but where does the magic happen? Where's our conscious mind? Self-awareness, thinking, remembering.
That, without a doubt, is the cerebral cortex, your brain's executive suite.
Even though it's so thin, like 2 to 4 millimeters.
It's thin, yes, but packed. Billions of neuron cell bodies, dendrites, glarula, blood vessels, no fiber tracts in the cortex itself.
But those convolutions triple its surface area.
Effectively, yes.
Yeah.
So it ends up being about 40% of your total brain mass.
In modern imaging, like PET scans, fMRI, they show specific areas for motor and sensory functions.
They do show localized domains, yes. But what's interesting is that higher mental functions, like memory and language, they're actually spread over large areas of the cortex, often overlapping.
So it really is the seat of the conscious mind.
It truly is.
So generalizing about how the cortex works, there are 4 main takeaways.
Right. First, it has 3 functional area types: motor, sensory, and association.
Second, contralateral control. Left brain, right body.
Mostly, yes. Each hemisphere handles the opposite side.
Third, lateralization. The hemispheres aren't identical in function.
Correct specialization.
And finally, no single area acts alone. It's all connected.
Conscious behavior involves the whole cortex, one way or another. It's a massive collaboration.
Okay, let's dig into the motor areas, mostly in the frontal lobe.
Primarily, yes. The primary motor cortex is right there in the precentral gyrus.
Home to the big pyramidal cells.
That's them. They let us consciously control precise, skilled, voluntary movements. They form the pyramidal or corticospinal tracts.
Okay, and this is where the motor homunculus idea comes in, right?
Exactly. It's a map, essentially.
A spatial map in the primary motor cortex.
But it's sort of upside down. Head at the bottom, toes at the top.
Right. And the really cool thing is how the size isn't based on the body part's actual size.
No, it's all about precision, how much fine control you need.
So like the face, tongue, hands, they get huge areas on this map.
That's why if you drew it, it looks like this, well, cartoon character. Huge lips, massive hands.
Tiny little legs. It's about the control, not the physical size.
Precisely. How much control we demand.
And this map also shows that contralateral control we mentioned. Left brain, right body.
Yes, exactly. But, we should add a bit of nuance there.
Okay.
The homunculus, it's maybe a bit of an oversimplification.
How so?
Well, it's not strictly one neuron maps to one muscle. It's more like individual neurons often send signals to multiple muscles.
Working together. Synergistic groups.
Exactly, synergistic groups, which has clinical implications too.
Like with a stroke.
Right. Damage to one spot paralyzes muscles on the opposite side, the contralateral side.
Makes sense.
But, and this is interesting, reflexive control might still work.
Oh, because that's a different pathway.
Yeah, it uses a different neural pathway, so it can sometimes remain intact.
Fascinating. Okay, just in front of that, we have the premotor cortex.
Right. This area helps plan movements.
How?
By selecting and sequencing basic motor actions into more complex tasks, like typing or playing an instrument.
And it uses sensory feedback to guide things.
Constantly. Coordinating multiple muscle groups, either together or one after the other. It's like the conductor for your everyday movements.
So if that's damaged, you might not lose strength.
No, not necessarily strength, but the skill is impaired.
You'd have trouble with those complex tasks,
Yeah.
like typing fast.
Exactly. And reprogramming that skill takes a lot of practice, like learning all over again.
Then there's Broca's area.
Yeah.
Traditionally linked to speech production.
Usually in the left hemisphere, yes.
Yeah.
Directing the muscles for speech.
But imaging shows it's active even when just planning other movements too.
That's right. It seems to be involved in sequencing actions more broadly, not just speech.
Interesting. And briefly, the frontal eye field.
Controls voluntary eye movements.
Okay, so that's commanding movement. But to move well, we need input, right? Sensory information.
Absolutely. Which brings us to the sensory areas. The primary somatosensory cortex is in the postcentral gyrus.
Receiving info from skin receptors and proprioceptors in muscles and joints.
Yes. And it allows for spatial discrimination, knowing exactly where you're being touched.
And it has its own map, the somatosensory homunculus.
It does. Also upside down, and also with disproportionately large areas for sensitive regions like lips and fingertips, reflecting all that sensory input.
And right behind that is the somatosensory association cortex.
Its job is integration. It takes the raw input from the primary cortex and helps you understand the object you're feeling.
Like its size, texture.
Exactly. If that area is damaged, you couldn't recognize objects just by touch.
Like feeling coins in your pocket but not knowing which is which.
Precisely that. You'd need to look at them.
Okay, moving to other senses. Vision.
Primary visual cortex is at the very back. Occipital lobe gets the raw input from the retina.
And the visual association area interprets it.
Yes, using past experiences, recognizing faces, objects.
And clinically, damage to the primary cortex causes blindness.
Functional blindness, yes. But damage to the association area is different. You can see, but you don't understand what you're seeing.
Wow. That's profound. Same idea for hearing. Primary auditory cortex for pitch and loudness.
And the auditory association area for perception, recognizing speech, music, a specific sound.
Right. And beyond sight and sound, there are areas for balance.
Vestibular cortex and the insula and parietal cortex.
Nope.
Olfactory cortex, part of the primitive rhinencephalon. Gustatory cortex, deep in the temporal lobe.
And even internal sensations, like a gut feeling or full bladder.
That's the visceral sensory area in the insula. It's this whole orchestra of specialized zones feeding into your conscious experience.
What's really wild is that most of the cortex isn't just primary sensory or motor areas. It's these multimodal association areas.
Yes. These are incredibly complex, interconnected regions. They get input from multiple senses and send output to multiple areas.
This is where it all comes together.
Pretty much. It's where individual perceptions get woven into a coherent whole. Information flows, sensory receptors, primary sensory cortex,
Yep.
sensory association cortex, multimodal association cortex.
And these areas give meaning to the information, stored in memory, help decide what to do.
Exactly. They're the glue making sense of your world.
Let's use that example from the text dropping acid in the lab.
Okay. You see it shatter, hear the crash, feel the burn, smell the fumes.
All separate inputs.
Right. They converge in these multimodal areas, along with maybe feelings of panic from the limbic system,
And it forms one seamless whole experience.
which then triggers your knowledge of what to do, run to the safety shower. Your premotor and motor cortex get the signal.
So these areas turn raw data into coherent experience and action. It's really what makes us us.
Absolutely. The anterior association area, the prefrontal cortex, that's the most complex bit. Your intellectual brain.
Involved in intellect, learning, recall, personality.
Yes, and working memory for abstract ideas, judgment, reasoning, planning, persistence.
And it develops slowly in kids, right? Needs social feedback.
Very much so. Clinically, damage here is devastating. Personality changes, loss of judgment, inhibitions. The executive functions go offline.
And the posterior association area.
Crucial for recognizing patterns, faces, knowing where you are in space, binding all those sensory inputs together.
Includes Wernicke's area for language understanding.
Correct. And then there's the limbic association area.
Adding the emotional weight, making memories stick.
Yes, working with the hippocampus. That feeling of danger with the assay, that's the limbic association area, making it significant.
And we touched on lateralization earlier, the division of labor between hemispheres.
Right. They look symmetrical, but they specialize. Cerebral dominance usually refers to the language hemisphere.
Typically the left, handling math, logic.
Usually. While the right is more involved in visual spatial skills, intuition, emotion, art, music.
The creative side complementing the logical left.
You put it that way, yeah.
So how does all this information zoom around the brain? Through the white matter.
Exactly. Deep to the gray cortex, the white matter is the communication network, the brain's superhighway system.
Made of myelinated fibers bundled into tracks for speed.
Yes, mostly myelinated. We classify them based on direction. First, association fibers.
Connecting different parts of the same hemisphere.
Correct. Keeping the left frontal talking to the left temporal, for example.
Okay, second type.
Commissural fibers. These connect corresponding gray areas of the two hemispheres.
Like the corpus callosum, the big bridge.
There's the largest one. It lets the two hemispheres coordinate, work as a whole.
And third.
Projection fibers. These run vertically. They either enter the cortex from lower areas or descend from the cortex down.
Tying the cortex to the rest of the nervous system. Body receptors, effectors.
Precisely. Structures like the internal capsule and corona radiata are formed by these projection fibers.
Okay, deep within that white matter, we find the basal nuclei.
Sometimes called basal ganglia, but nuclei is better since they're in the CNS.
Islands of gray matter, hidden away.
They're a cluster of distinct structures, caudate, putamen, globus pallidus, functionally linked with others, like the subthalamic nuclei and substantia nigra. Think of them as a team of movement editors.
Editors, how so?
They get input from all over the cortex and other subcortical areas. They don't directly control muscles.
But they influence the movements directed by the primary motor cortex.
Exactly. They filter out incorrect or inappropriate responses. They help select the best response to pass back to the cortex.
So they help start and stop movements. Monitor intensity, like arm swinging when walking.
Yes, those slower, stereotyped movements. And crucially, they inhibit unnecessary movements, keeping things smooth and intentional.
And when they malfunction, that's where we see things like Parkinson's or Huntington's.
Correct. Their dysfunction is central to those disorders, really highlighting their importance in motor control.
Okay, let's move deeper to the diencephalon, the central core of the forebrain.
Made of 3 paired structures around the third ventricle. Thalamus, hypothalamus, epithalamus.
The thalamus, the inner room, makes up most of it, about 80%.
Yes, and it's truly the gateway to the cerebral cortex.
The grand central station.
That's a perfect analogy. Pretty much all information heading to the cortex funnels through the thalamus.
All sensory info. Touch, pressure, pain.
All afferent impulses converge there. They get sorted, edited. The thalamus gives you that crude sense, pleasant, unpleasant, before the cortex does the fine detail.
And motor signals pass through, too, from cerebellum basal nuclei.
Yes, helping direct motor activity. It's involved in sensation, motor control, cortical arousal, learning, memory, a major hub.
So if the thalamus is the relay station, what about its tiny neighbor, the hypothalamus? What makes it so mighty?
The hypothalamus. Small but incredibly powerful. It's the main visceral control center, absolutely vital for homeostasis.
Controlling things without us even noticing.
Pretty much. Its roles are so diverse. It controls the autonomic nervous system, blood pressure, heart rate, digestion, pupil size.
Wow.
It initiates physical responses to emotions. It's at the heart of the limbic system. Pleasure, fear, rage, sex drive.
It's the body's thermostat, regulating temperature.
Yes, initiating sweating or shivering. It regulates food intake, hunger, satiety.
Water balance and thirst, too.
Via osmoreceptors triggering ADH release. It regulates sleep-wake cycles with its suprachiasmatic nucleus, the biological clock.
And controls the endocrine system.
Big time. Releasing or inhibiting hormones for the pituitary, plus producing ADH and oxytocin itself. It's your internal autopilot.
Astonishing. And if it's disturbed, emotional imbalances, failure to thrive in kids.
Exactly. Its influence is profound.
And the last part of the diencephalon, the epithalamus.
The most dorsal part forms the roof of the third ventricle, contains the pineal gland,
which secretes melatonin for sleep-wake cycles.
That's its main job, regulating that natural rhythm.
All right, next stop. The brainstem. Tiny, only 2.5% of brain mass, but critical for survival.
Absolutely. Midbrain, pons, medulla oblongata, it handles rigidly programmed automatic survival behaviors, breathing, heart rate.
And it's a pathway for fiber tracks connecting higher and lower centers.
Plus, its nuclei are associated with 10 of the 12 pairs of cranial nerves innervating the head.
Let's break it down, the midbrain.
You've got the ventral cerebral peduncles containing descending motor tracts. The cerebral aqueduct runs through it.
Surrounded by the periaqueductal gray matter for pain suppression, fight or flight.
Right. And dorsal structures, the corpora quadrigemina, superior colliculi for visual reflexes.
It's tracking things.
Yeah. Inferior colliculi for auditory relays, the startle reflex.
And deep inside, substantia nigra, Parkinson's connection.
Yes, dopamine production, degenerates in Parkinson's, and the red nucleus involved in limb flexion.
Okay. Next, the pons, the bridge.
Literally. Mostly conduction tracks, connecting higher centers to the spinal cord and, crucially, connecting the pons to the cerebellum via the middle cerebellar peduncles.
Associated with cranial nerves, V6, V6-7, helps regulate breathing rhythm.
Yes, works with the medulla on that smooth breathing pattern.
And finally, the medulla oblongata, most inferior part, blends into the spinal cord.
At the foramen magnum, yes. Ventrally, you see the pyramids formed by the large motor tracts.
And the decussation of the pyramids happens here, the crossover.
Just above the spinal cord junction. That's why the left brain controls the right body.
And the medulla houses vital autonomic centers.
Absolutely critical ones. Cardiovascular centers adjusting heart rate, vessel diameter, respiratory centers controlling breathing rate and depth.
Plus centers for vomiting, hiccuping, swallowing, coughing, sneezing. The real survival stuff.
Definitely this survival hub.
Last but not least for this section, the cerebellum, the small brain.
Second largest part after the cerebrum. Tucked under the occipital lobes, dorsal to the pons, and medulla.
And its key role is coordination. Smooth movements, agility.
Precisely. It provides the exact timing and patterns for skeletal muscle contraction. Think driving, typing, playing sports. That's the cerebellum ensuring it's all fluid and coordinated.
Anatomically, two hemispheres connected by the vermis. Surface is folded into folia.
Yes, very convoluted. And inside that distinctive white matter pattern, the arbor vitae.
The tree of life looks just like it.
It really does. And it connects to the brainstem via 3 pairs of cerebellar peduncles, superior, middle, inferior. Those are its communication lines.
And unlike the cerebrum, its fibers are ipsilateral, connecting to the same side of the body.
Mostly, yes. That's a key difference from the cerebrum's contralateral control.
So how does it process things? It gets intent from the cortex.
Receives motor intent, yes. Then it gathers info from proprioceptors, visual system, equilibrium pathways.
Figures out the best way to coordinate the muscles.
Calculates the optimal plan. Then it dispatches a blueprint back up to the motor cortex to fine-tune the movements.
It's amazing, all happening subconsciously when you just reach for a coffee cup.
You have no awareness of it. Injury here doesn't cause paralysis, but loss of muscle tone, clumsy, uncoordinated movements, like intention tremors.
And emerging research suggests it does more. Thinking, language, emotion.
There's growing evidence for cognitive roles, too. It's more than just a motor coordinator.
Okay, incredible foundation. So how do these structures work together for higher functions? And how is it all protected?
Well, take language. Such a complex function. It uses almost all the association cortex, usually on the left side.
Broca's area for speaking writing. Wernicke's for understanding.
Classically, yes. Damaged Broca's, you struggle to produce language, though you understand it. Damaged Wernicke's, you can speak, but it's word salad, makes no sense, and you struggle to understand.
And these work with basal nuclei as a language system.
Right, and the right hemisphere adds the emotional context, tone of voice, gestures. It's a complex interplay.
And memory. storing and retrieving info.
Yeah.
Essential for learning, consciousness.
Our lifetime of experiences, really. We talk about short-term memory, STM.
Limited capacity, like 7 or 8 items. A phone number.
And long-term memory, LTM, which seems almost limitless, so we can forget things.
What helps transfer info from STM to LTM?
Several factors. Emotional state, strong emotions, norepinephrine release can burn things in, rehearsal, repetition helps.
Association, linking new stuff to old stuff.
Very effective. And automatic memory, some things just stick without effort. Memory consolidation involves the hippocampus and temporal cortex, fitting new info into existing knowledge.
And clinically, hippocampal damage causes amnesia.
Yes. Anterograde amnesia can't form new memories, or retrograde amnesia, losing past memories.
How do we even measure all this brain activity? EEGs, brain waves.
Exactly. The electroencephalogram records the continuous electrical activity of neurons, used to diagnose epilepsy, sleep disorders, determine brain death.
And there are different wave types, alpha, beta, theta, delta.
Based on frequency, yes. Alpha waves are the relaxed, awake, idling brain.
Beta waves.
Mentally alert, concentrating, like listening now.
Beta.
More common in children. Unusual in awake adults might indicate issues.
And delta.
High amplitude, deep sleep waves. If seen in awake adults, indicates brain damage. But these waves show the brain's underlying rhythm. And critically, a flat EEG, no spontaneous waves, means brain death.
Which brings us to epilepsy, those torrents of electrical discharge.
Seizures.
Exactly. Can range from absent seizures, brief loss of consciousness, to tonic-clonic seizures, the severe convulsive type.
And some people get an aura beforehand, a sensory warning.
Yes, a strange smell, visual distortion. It could be a helpful warning.
This all ties into consciousness, right? Perception, voluntary movement, higher processing. Yes, clinically it's a continuum. Alertness, drowsiness, stupor, coma. Current thinking is consciousness involves large cortical areas working together simultaneously.
It's holistic, interconnected, not just one spot.
Right. And fainting, syncope, is temporary loss from low blood flow. Coma is much more serious, significant unresponsiveness, lower brain oxygen use than even deep sleep.
Okay, speaking of sleep, it's partial unconsciousness you can be aroused from, unlike coma.
Correct. 2 main types. NREM, non-rapid eye movement sleep.
Stages 1 to 4. Slow wave sleep, restorative.
That's the idea, physical restoration. Then there's REM sleep, rapid eye movement.
Eyes move, muscles inhibited, most dreaming happens. Emotional processing.
Believed to be important for emotional stability, consolidating memories. These alternate in a circadian rhythm timed by the hypothalamus.
The suprachiasmatic nucleus again.
Yeah.
And the preoptic nucleus inhibits the RAS, the arousal system.
Yes, helps induce sleep. And other hypothalamic neurons release orexins, the wake-up chemicals.
Which ties into narcolepsy. Abrupt REM sleep onset, often linked to low orexin.
Exactly, and insomnia is the chronic inability to get adequate sleep.
So what does this all mean for protection?
Yes.
How is this vital, delicate tissue shielded?
4 main layers of protection. Bone, the skull, meninges, the membranes, cerebrospinal fluid, the cushion, and the blood-brain barrier, the gatekeeper.
Let's take the meninges first. 3 connective tissue membranes.
External to the CNS, outer dura mater, tough mother, strongest layer, forms dural septa like the falx cerebri.
Which limit brain movement.
Exactly. Middle arachnoid mater, loose covering, subdural space separates it from dura. Web-like extensions connect to the inner pia mater. Subarachnoid space beneath has CSF blood vessels.
And arachnoid granulations absorb CSF back into the blood.
Yes, into the dural venous sinuses. Finally, the pia mater, gentle mother, clings tightly to every fold of the brain.
And inflammation of these is meningitis. Can spread to the brain.
Yes, causing encephalitis. Very serious.
Okay, protection layer 2. Cerebrospinal fluid, CSF.
That watery broth in and around the brain and cord, its main function is buoyancy.
Reduces brain weight by 97%. Prevents self-crushing.
Incredible, isn't it? It also cushions against trauma and helps nourish the brain. Shock absorber and delivery system.
Formed by choroid plexuses in the ventricles.
Yes, capillaries enclosed by ependymal cells. They actively modify blood filtrate to create CSF. About 500 milliliters form daily. Total volume is 150 milliliters, so it turns over fast.
Circulates through ventricles, out to subarachnoid space, absorbed by arachnoid granulations.
That's the path.
And clinically, if that path is blocked, hydrocephalus. Water on the brain.
Yes, CSF builds up. In babies, the head enlarges. In adults, the rigid skull means pressure builds, damaging the brain.
Treated with shunts to drain the fluid.
Often, yes, relieves the pressure.
And the final protection, the blood-brain barrier.
A crucial selective barrier. Very tight junctions between capillary endothelial cells, like a strict bodyguard.
Letting good stuff in, keeping bad stuff out.
Highly selective, allows glucose, oxygen, essential amino acids, fat-soluble things like alcohol, nicotine.
But blocks, wastes, proteins, most drugs.
Exactly. Maintains a stable brain environment. It's absent in a few spots, like the vomiting center and hypothalamus, which need to monitor the blood directly.
And it's not fully formed in newborns, making them more vulnerable.
That's right. Develops over time.
But even with all this protection, things can go wrong. Traumatic brain injuries, TBIs.
A major cause of accidental death. A concussion is a temporary alteration in function.
But multiple ones cause cumulative damage. Long-term issues.
We now know that's a serious concern, yes. A contusion is worse: brain bruising, permanent damage. Severe brainstem contusions often cause coma by damaging the arousal system, RAS.
Bleeding in the brain. Subdural or subarachnoid hemorrhage.
Yes, ruptured vessels cause bleeding, increasing pressure, compressing tissue, often needs surgery.
And cerebral edema. Brain swelling.
Can happen with injury or on its own. Can be fatal.
Then there are strokes. Cerebrovascular accidents, CVAs.
The most common nervous system disorder, a leading cause of death, happen when blood circulation to a brain area is blocked.
Ischemia, leading to tissue death.
Exactly. Usually caused by a blood clot, but bleeding can also cause strokes. Survivors often have hemiplegia, paralysis on one side, plus sensory or speech deficits.
And TIAs, transient ischemic attacks, mini-strokes.
Yes, temporary episodes, reversible ischemia, crucial red flags, warning of a possible major stroke.
And glutamate, the neurotransmitter. It actually makes stroke damage worse.
Tragically, yes. It acts as an excitotoxin after the initial blockage. Overstimulates neurons to death.
Wow. Treatment includes TPA, the clot buster, if given quickly.
Time is critical.
Okay, let's touch on degenerative brain disorders. Alzheimer's disease, AD.
A progressive disease causing dementia. Affects many over 65. Hallmarks are senile plaques.
Beta amyloid clumps outside neurons.
Yes, and neurofibrillary tangles inside neurons involving tau protein. Also, loss of acetylcholine-producing neurons impacting memory.
Parkinson's disease usually hits in 50s, 60s. Degeneration of dopamine neurons in substantia nigra.
Correct. Leads to overactive basal nuclei. Symptoms include resting tremor, pill rolling, shuffling gait, stiff facial expression, slow movement.
Treatments like L-DOPA, deep brain stimulation, can help manage symptoms.
They can, yes. No cure yet.
Huntington's disease. Fatal. Hereditary. Mutant Huntingtin protein.
Yes, accumulates and causes degeneration of basal nuclei and cortex. Leads to wild, jerky movements, chorea, and mental deterioration. Devastating.
And diagnosing these often involves advanced imaging. CT, MRI, PET scans.
Absolutely. They help identify tumors, lesions, dead tissue. Cerebral angiography visualizes blood vessels, useful for stroke risk. TIAs.
Okay, shifting focus slightly now to the spinal cord. More than just a cable.
Definitely. Two-way conduction pathway, major reflex center.
Yeah.
Extends from the skull's foramen magnum down to about L1 or L2, just below the ribs.
Protected like the brain. Bone, meninges, CSF.
Yes. Spinal duramater is single-layered, not stuck to vertebrae. Epidural space outside it has fat, veins. CSF is in the subarachnoid space.
Which allows for lumbar puncture of the spinal tap.
Right. Done safely below L3 where the cord ends. Needle pushes nerve roots aside. Used to get CSF for diagnosis.
Inferiorly, the cord tapers to the conus medullaris.
A cone shape, yes. Then the filum terminale anchors it to the coccyx.
And the cauda equina, the horse's tail.
That's the collection of lower spinal nerve roots extending down because the vertebral column grew faster than the cord.
Plus cervical and lumbar enlargements where limb nerves arise.
Handling all the innervation for arms and legs.
In cross-section, gray matter is H-shaped inside, white matter outside.
Correct. Gray matter horns. Dorsal, ventral, sometimes lateral, connected by gray commissure around the central canal.
Dorsal horns have interneurons, relaying info.
Mostly. Ventral horns have somatic motor neurons controlling voluntary muscles. Their axons exit via ventral roots.
Lateral horns, autonomic motor neurons.
For involuntary functions, yes. And sensory fibers enter via dorsal roots, with cell bodies in the dorsal root ganglia outside the cord.
The surrounding white matter has columns, funiculi, with ascending and descending tracks, the information highways.
Exactly. Dorsal, lateral, and ventral funiculi carrying signals up and down.
Spinal cord trauma. Devastating consequences, dorsal root damage.
Loss of sensation or paresthesias, tingling, numbness.
Ventral root or horn damage.
Paralysis. Motor commands can't get through.
And there are two types of paralysis, flaccid versus spastic.
Right. Flaccid paralysis, nerve impulses don't reach muscles, they atrophy. Spastic paralysis. Upper motor neuron damage, muscle still healthy but no voluntary control, often stay contracted.
Complete cord transection, total loss, below injury.
Yes. Paraplegia if between T1L1, lower limbs. Quadriplegia if in cervical region, all four limbs.
Yes.
Usually followed by spinal shock initially.
And diseases like polio, virus destroying ventral horn motor neurons.
Leads to paralysis, yes. And ALS, Lou Gehrig's disease, progressively destroys ventral horn motor neurons and pyramidal tracts.
Leading to loss of speech, swallowing, breathing, cognitive function often spared. Heartbreaking.
Truly tragic.
Okay, let's talk about the pathways themselves. What makes these neural highways so efficient?
4 key characteristics. First, decussation. Most pathways cross over.
Second, relay. Chains of neurons passing the signal, usually 2 or 3.
Third, sympathetic. Precise spatial mapping reflecting the body map.
Your fourth, symmetry. Paired pathways on each side.
These allow rapid, precise information flow. Ascending pathways carry sensory info up.
Through 3 neurons. First, second, third order.
Generally, yes. First order. Receptor to CNS. Second order. Relay within CNS to thalamus or cerebellum. Third order. Thalamus to somatosensory cortex for conscious awareness.
And the 3 main somatosensory pathways are?
The dorsal column medial limnoscal pathways. For precise touch, vibration. Decussate in the medulla.
The spinothalamic pathways. For pain, temperature, crude touch, pressure. Decussate in the spinal cord.
And the spinocerebellar pathways convey muscle tendon stretch info to the cerebellum for coordination, not conscious sensation.
Okay, and descending pathways, motor commands going down.
Delivering efferent impulses involve upper motor neurons from brain and lower motor neurons in spinal cord directly innervating muscle.
We split them into direct pyramidal and indirect pathways.
Correct. Direct pathways originate mainly in the precentral gyri. Regulate fast, fine, skilled movements like writing.
Indirect pathways, more complex, multisynaptic.
Yes. Involved in regulating axial muscles for balance, posture, coarse limb movements, head, neck, eye movements for tracking, like the background support system.
Finally, let's look at development and aging of the CNS, a lifelong process.
Starts incredibly early with that neural tube forming gray matter plates, white matter tracts. Interesting point. Gender-specific areas appear prenatally?
Like hypothalamic nuclei being larger in males if testosterone's present.
During a critical window, yes.
And maternal exposure to toxins, radiation, drugs, alcohol infections can severely harm fetal CNS development.
Absolutely. Things like rubella causing deafness, smoking, reducing oxygen. Prenatal care is so crucial.
This is where we see congenital issues too. Cerebral palsy.
Neuromuscular disability, often from oxygen lack at birth, affects movement.
And encephaly. Brain doesn't develop properly.
Incompatible with life, tragically. Spina bifida. Incomplete vertebral arches. Ranges from mild occulta, often no symptoms, to severe cystica where the cord itself might protrude. And a key insight.
Folic acid deficiency.
Yes, a major cause. Why prenatal vitamins with folic acid are so important.
And the hypothalamus matures late, affecting temperature regulation in preemies.
That's right, they often need incubators. Neuromuscular coordination also progresses predictably. Head to toe, proximal to distal, following myelination patterns.
Head control before leg control. Shoulder control before finger control.
Exactly.
What about the aging brain? Some decline is normal.
Brain weight volume decreases after young adulthood as some neurons die, but remaining neurons can adapt, form new connections.
Cognitive declines after 70. Spatial ability reaction time. But math? Verbal skills often stable.
That's generally true. And importantly, some dementia is reversible, caused by treatable things like drug side effects or poor nutrition.
That offers some hope.
It does.
But factors like chronic alcoholism, repeated head trauma, boxing, contact sports can accelerate deterioration.
Sadly, yes. They take a significant toll beyond normal aging, a reminder of how delicate yet resilient our brains are.
Wow. We've covered an incredible amount of ground today, an amazing journey through the central nervous system.
From its embryonic start through all the protective layers.
To the complex functions of the hemispheres, diencephalon, brainstem, spinal cord.
And those vital neuronal pathways connecting everything.
What's truly remarkable, I think, is understanding these foundations. It's key to appreciating the body's vast capabilities and its vulnerabilities.
Absolutely. This deep dive into Human Anatomy and Physiology, 10th edition, really shows how interconnected everything is. It forms the very essence of, well, you.
We really hope this deep dive has given you that shortcut to being well-informed and maybe sparked quite a few aha moments about your own incredible internal world.
It's been fascinating to explore it with you.
Thank you so much for being part of our last-minute lecture family. Until next time, keep exploring, keep learning, and stay curious about the marvels within you.